Thank you so much for the invitation to speak. Great. So a few disclosures. My intellectual conflict is that I do believe in treatable traits, and one of the advisory board members is trying to develop a companion diagnostic. So we just saw this really elegant figure from I think a highly influential paper that was conceived of and led by David Maslov and John Marshall, who you'll hear probably a lot more about this next. But Dr. Thompson really highlighted how perhaps we could, perhaps we could envision kind of this red dot as a shared trait that we could see across multiple syndromes. And I think we've all experienced this as ICU clinicians, where we might have a severely injured patient who we did not think was infected, but was manifesting dramatic vasopressor-dependent shock and fever and inflammation, and wondered whether we should adapt some of our sepsis treatments. So maybe we could term those treatable traits once we find the treatments that work. And I really liked this concept that was put out in a review of treatable traits within obstructive lung disease. But trying to break down, we know that there's tremendous heterogeneity in obstructive lung disease, and yet perhaps patients that share certain features like eosinophilic inflammation might be predicted to respond differently to therapies. So when we're talking about treatable traits, we're looking for traits that are clinically relevant. They have to mean something. So whether that's that they respond differently to treatment or they have a different prognosis, both could be useful. They have to be measurable, identifiable. Eventually you have to operationalize those and say, what does a positive troponin mean? And is that context specific? And of course, they have to be treatable. So we'll go through a couple of aspirational examples from other complex traits and ask what we could adapt. I'm deliberately going to eschew cancer a little bit because I think that's a very different example that has pathology to guide them. We'll look at our successes in sepsis and ARDS and ask how close we're getting. And what could we do to accelerate future success? So just last week's New England Journal has the new poster child for precision medicine. And this is an incredibly scientifically elegant description that leveraged the knowledge that, especially in the United States, we recognize that our patients with African ancestry have a huge increased risk for chronic kidney disease compared to patients of European ancestry. And so leveraging genetics, we were really able to pinpoint this risk to variants on the APOL1 gene. And these variants were coding, they changed the protein sequence, and they were private to African population. So up at the top in red is kind of showing you the distribution of these, where these genotypes are found. And like I said, a huge risk. So odds ratios of 29 for some of these traits, especially traits that caused heavy proteinuria. So that led to a lot of investigation. Why would APOL1 be involved in kidney disease? It's a channel. It changes your potassium flux. Knowing that, scientists were able to do small molecule drug screening, looking for how could they modify the APOL variant. They then did elegant animal studies. They could actually use CRISPR-Cas to knock in the human variants and use that as a model for their drugs and ask which drugs could influence this mouse. And it culminates in a 15-patient trial, genotype-directed trial, phase 2, single arm, all patients got the drug. And they had a great readout of proteinuria. And showing you here the 13 patients who met their a priori adherence criteria, 12 of the 13 had dramatic reductions in their proteinuria. One patient did not respond, and they're trying to figure out why that is. But this is a genotype-guided trial with a really solid rationale with beautiful biologic. And I think worth noting, isn't it nice to see this type of effort and money put into a disease that affects our African ancestry parts of the world, which have historically been underfunded. Our cardiologists have been able to leverage genetics very effectively. So they focused on LDL and could use that as a quantitative trait, which then has a lot of power statistically to do genetic screens and say, what are the genes associated with LDL? And look at how many of these have kind of been converted to therapies. So we now have PCSK9 inhibitors because we understood that patients who are PCSK9 null have dramatically lower LDL. There are new gene targets looking to silence the sortilin gene. ApoE is another target. So how were they able to make these advances? Because they had defined causal traits. They used causal inference to show that LDL mattered for MI risk and MI mortality. It was quantitative and therefore statistically powerful and easy to bring back to the clinic so we could measure LDL on our patients and develop those cut points. And it really clarified their therapeutic goals. Lots of other examples in other complex diseases. But again, if we think about what we're trying to achieve, we would like in sepsis and ARDS to find our causal drivers. We'd like to know what quantitative traits could help us get to those causal drivers. And then, maybe there are some tools that would at least help us sort our patients, whether we're sorting by prognosis or by treatment. So our group used a small genetic screen, really, in retrospect, and found a fairly consistent relationship between a variant in the interleukin-1 receptor antagonist gene and lower risk for both ARDS and sepsis death. It seemed to be a functional variant that caused you to make IL1RA faster, and that could subsequently perhaps suppress your IL-1 beta. And of course, that was interesting because people had wondered about blocking interleukin-1 beta with synthetic IL1RA in sepsis and had done several phase 3 trials that did not have a large effect, not a large average treatment effect, maybe we'll say. But we wondered, seeing this heterogeneity, both genetic and in the plasma proteins, maybe this effect might be different in patients with different IL-1 dysregulation. So I was fortunate to collaborate with Stephen Opel, one of the PIs from that sepsis phase 3 trial, who still had samples left over, 20-year-old samples from the freezer. We were not able to genotype them. We tried to do whole genome amplification. That didn't really work, but we were able to measure plasma IL1RA and plasma IL1 beta, and we identified a statistical interaction between the mortality effect of this randomized drug and your risk for mortality, and where you were in terms of your IL1RA expression. And whether we did that as a single cut point or looked across multiple cut points, looking at deciles of IL1RA, we saw this. We didn't think we were just identifying severity of illness and that we did not appreciate the same interaction for Apache by itself, looking at different Apache strata. And this was our rationale to go ahead and do a stratified analysis, stratifying by IL1RA expression. And hopefully you can see here in red are the patients with high plasma IL1RA who have a mortality benefit from being randomized to recombinant IL1RA, whereas in blue are the patients with low expressing baseline IL1RA who, if anything, seem to do worse when they're randomized to recombinant IL1RA or anakinra. So is this a treatable trait? Do we have high plasma IL1RA as a treatable trait? I would say not yet. So certainly we need first a qualified IL1RA assay that works the same in different contexts. We definitely need prospective replication in a contemporary setting. That was 20 years ago. And I'd argue this is good rationale to retest IL1RA. And adding to that might be the Save More trial that in floor patients with COVID, again, seemed to appreciate an effect. So I'm advocating for more testing. If we think where we are in sepsis, so we've done very well on the classification side, and several different groups have come up with whole blood gene expression classifiers. So these are looking at the messenger RNA expressed by all of your circulating white blood cells. The Knight and Davenport group reported the sepsis response signatures. The Mars group from the Netherlands had Mars 1 through 4. And Tim Sweeney's group integrated a lot of large data sets, including the other twos, and came up with 3 clusters that seem to replicate in a number of populations. So interestingly, the sepsis response signature has been tested at least in one subgroup of the VANISH trial. David Ancliffe and Tony Gordon went back to their VANISH patients who still had a PAX gene tube or whole blood gene expression, and were able to show that SRS2, which is typically the very low mortality group, when randomized to hydrocortisone in red, had a much higher mortality, suggesting there is harm in these SRS2 patients from being randomized to hydrocortisone. Now, that was a randomized study. However, it is a very small number of patients because it was a subset, and with small numbers, we always have to be worried that our results might be more extreme than truth. So I would say again, maybe this is a treatable trait. I'd like to see it replicated in a larger study to be more confident that this mortality difference, which is so large, is repeated. We heard about ARDS subphenotyping and hypoinflammatory, hyperinflammatory, or of course, Dr. Boss's group that did the reactive and uninflamed. These 2 phenotyping schemes share a lot of similarities, with plasma protein expression being the real drivers. And certainly Dr. Calfee's group have shown heterogeneous treatment to randomized PEEP, to randomized fluid strategy, and to randomized simvastatin. So is hyperinflammatory ARDS, is that a treatable trait? Again, it's measurable. We think it's significant based on both this differential effect and the mortality. But can we prospectively validate it now? And so I think there are several efforts, including the FIND study, to try to do just that. So what do we need to do going forward? Like I said at the outset, I'm a believer and a Ted Lasso fan. But I really think we need to find those quantitative traits. Those numeric traits are just so valuable in helping us to find those biological signals. Wouldn't it be great if we had an LDL for ARDS or for sepsis and could really clarify some of our therapeutic efforts? Once we have those traits, we really need to test their causality and or their ability to consistently classify patients. I probably didn't stress this enough, but a lot of the advances that were made in asthma, in cardiovascular disease, leveraged the randomized trials that they already had. That's really our only way to test what is treatable when you can use randomization. So I would really recommend that we always try to include biosamples into the majority, if possible, of our patients enrolled in trials. Thank you.